organizing committee for the opportunity to present my work at this wonderful conference. My work named is Computer-Aided Design of Novel Tspo Ligands, Potential Neuropsychotropic Agents. This work was carried out in Zakusov Research Institute of Pharmacology in Moscow, in Russia. According to World Health Organization for 2019, the central nervous system diseases were diagnosed in approximately 1 billion people on the planet. And the most common diseases in this area is anxiety, depressive disorders, neurodegenerative diseases, and the economic losses associated with this type of diseases, it's about $1 trillion. The COVID-19 pandemic has significantly contributed to the prevalence of such diseases. For example, in 2020 alone, the prevalence of anxiety and depressive disorders increased by almost 30% due to COVID-19. It is well known that the major limitation of use of non-neuropsychotropic drugs is the large number of their side effects. For example, in benzodiazepines, as the known anxiolytics, for example, diazepam, the side effects are sedative, muscle relaxant, amnestic, and addictive effects. Among the famous antidepressants, such as selective serotonin reuptake inhibitors, for example, fluoxetine, there are also a lot of side effects, such as convulsions, gastrointestinal disorders, aggression, and withdrawal symptoms. The search for new mechanisms, the new neuropsychotropic drugs, which would be safe, fast, and efficient, is the great challenge. One of the promising biotarget for creation of the novel neuropsychotropic agents is translocated protein with a mass of 18 kilodaltons. Until 2006, this protein was known as peripheral benzodiazepine receptor. Translocated protein located on the membrane of mitochondria in the human brain. And the key role of this receptor is the transport of cholesterol into mitochondria. That is the rate-limiting step in biosynthesis of neurosteroids. Neurosteroids have their own binding site on the GABA-A receptor. They act as positive allosteric modulators. And this binding site is different from benzodiazepine binding site. So neurosteroids act as safe and fast neuropsychotropic agents. Unfortunately, neurosteroids couldn't be used as drugs on their own because they're metabolic unstable. And so the activation of biosynthesis of neurosteroids by Tspo Ligands is a promising approach to create the fast, safe neuropsychotropic agents. To create the new Tspo Ligands, we have the following strategy. At first, we collected the database of known Tspo Ligands, which could allow us to create the pharmacophore models of these ligands to receive the new group of these compounds. And on the next stages, we plan to make the docking optimization and ADMET optimization of novel ligands to select the molecules for their synthesis. On the first stage, we collected the database of Tspo Ligands. This database have about 100 molecules, and the example of these molecules are on this slide. And we found that the most Tspo Ligands have the similar pharmacophore structure. They have flat aromatic heterocycle, aromatic substituent, and amide lipophilic substituent, and hydrogen bond acceptor. Based on this speculative model, we created the group of pyrrolopyrazine potential ligands, and on the next stage, we analyzed the calculated pharmacophore model, which was created using the database of Tspo Ligands and using the Schrödinger phase program. The most probably hypothesis of the pharmacophore model was this, which contains two aromatic rings, the hydrogen bond acceptor and hydrophobic group. Using this precision pharmacophore model, we selected the molecules in our group for further development. The next stage was the docking studies. We used the structure of mouse Tspo in the complex with its native ligand PK11195 from protein data bank. On this picture, you can see the position of this native ligand in the translocator protein. And using Schrödinger Glide software, we made the docking studies, and the molecules with parameter docking score of no more than -7 were selected in this docking optimization. In this table, you can see the structures that we selected. The docking score parameter was from -7 to -10. The K interaction of ligands with the protein were PP staking with tryptophan 107 and tryptophan 143, and also the number of hydrophobic interactions with several amino sequences. Here is the example of docking of molecule GML1 in the Tspo binding site. You can see here the PP staking of pyrrole ring with tryptophan 143 in hydrophobic interactions. And on this slide, you can see the interaction of GML12 with Tspo binding site. And here we can see PP stakings of aromatic rings with tryptophan 107 and tryptophan 143, and also in 3D view. And the next stage of the optimization was ADMET optimization. We wanted to select the molecules with good pharmacokinetic and toxicology parameters. The main analyzed ADMET parameters were molecular weight, dipole moment, lipophility log P, penetration the blood-brain barrier log BB, and also Lipinski rule of 5, Jorgensen rule of 3, oral availability, Ames test, and acute toxicity in rats. Here you can see the table of these results. ADMET parameters were calculated using quick probe software from Schrödinger and ADMET lab. And the most compounds with good docking score parameter had also very good ADMET parameters, except 2 molecules. And so we selected these molecules for further synthesis to analyze their Tspo properties. Here's the synthetic scheme of new compounds, and the synthesized compounds were studied for their anxiolytic activity at first using animal models. It is open field test in mice and elevated plus maze test in mice, which showed the anxiolytic activity. And on this scheme, you can see the structure-activity relationship results. I will stop here, and I want to say that the main results is that the active compounds have the better results in docking score parameter and also in lipophility parameters log P and blood-brain barrier penetration log B parameter. And so we can see that these parameters can be used to assess the ability of compounds to be active. The lead compounds are presented on this slide. It is GML1, GML3, 11, and 102. These compounds were active in different doses in open field test, in elevated plasmase test, and also in tail hung test, which showed anxiolytic and antidepressive activity of new compounds. And the first two compounds were selected for the preclinical studies. GML1 was selected as potential rapid anxiolytic and GML3 as potential fast antidepressant. We studied their affinity against translocator protein using radioligand binding studies with radioligand PK119.5. This research was carried out in Sarep company, and we found that our molecules had very good affinity parameters. It is 15 and 500 nanomoles of their IC15 parameter. And it is important to see that this IC15 parameters has a very good correlation with their docking score parameters. Compound GML1 studied in full preclinical studies as rapid anxiolytic in the different departments of our institute. We found that this compound has very high anxiolytic activity in different animal tests. Also, it has had antidepressant activity and nootropic activity. It is very important because it is well known that the famous anxiolytics, for example, benzodiazepines, have very negative effect on the memory. And our compound have positive influence on the memory. We proved the Tspo mechanism of our compound using inhibitor analysis with Tspo blocker PK111.95 and with inhibitors of neurosteroid biosynthesis. These compounds prevent the effects of our molecule that prove the Tspo mechanism. In the dosage form department, a tablet dosage form of GML1 has been developed. And in pharmacokinetic department was made the full pharmacokinetic studies of this compound. We found that it have very good absolute bioavailability. And we studied all safety parameters of GML1. We found that this compound didn't have any toxic effect on mice and rats. It didn't have any immunotoxic effect, and it hasn't embryotoxic, fetotoxic, teratogenic, mutagenic, and carcinogenic effects. And so now compound GML1 is ready for clinical trials as rapid anxiolytic with pro-cognitive effects. And at the end, I would like to thank all the departments that take part in the development of Tspo Ligands and GML1. It is the Department of Pharmacogenetics, pharmacokinetics, toxicology department of our institute, and departments of dosage form. And I would like to say that our project was supported by Pharma 2020 project. And thank you for your attention.
Thank you for you. I see one question in our chat. Why different animals? Just a moment. Why different animals are used for different behavior tests?
Well, we used mice and rats in all tests. And I don't understand why the question is about different animals. Here we used mice, different lines of mice, because, for example, bulb mice are used in open field test because they are very sensitive in this test. Okay.
Why different strains?
What?
I read next question from Sachin. I think in section 6 question. Why different strains?
Different types of mice.
Maybe.
Well, every type of mice is used in different tests because they are sensitive each in every test. And so we used different mice types in different animal tests.
Okay. And I see next question. Why you have selected TM2, TM4, and TM5 region for your molecular docking study? First question. Second question, what about TMY and TM3 region? And next question, please explain the significance of Jorgensen rules of 3.
Well, in our docking studies, we used the active site of Tspo protein, and TM2, TM4, and TM5 form the binding site of this receptor. So the docking studies were elucidated in this site. And the next question.
Next question, what about TMY and TM3 region?
Well, these regions don't play the significant role in the binding with ligands, and so they don't use. And the significance of Jorgensen rule is very close to Lipinski rule of 5. It is explain the number of hydrogen bond donors, acceptors, and some other parameters of molecules that should be correspond to prospect molecules.
Thank you.